Dr. Javeria will be chairing today's session. Co-chair Dr. Rumina Ghazi from Shaukat Khana Memorial Cancer Hospital. And moderating the session today is Dr. Shavana Shahid from Shifa International Hospital. Thank you. Over to you, Dr. Shavana. Dr. Javeria, would you just like to say a few words while we wait for just a few minutes to get everybody in the room?
Assalamu alaikum. Thank you everyone for being here. So I think this session is focused on a very important topic about editing. So a lot of us, molecular pathology I think is unique in this sense. We are at the Pakistan Association of Pathology conference, but molecular pathology uniquely brings together scientists and clinicians. So I think this is unique about our forum, and that's why we have quite innovative program and blossoming of new ideas. So this is something that I'm very, very excited about. So for the longest time I've been working in hematopathology, and I've been attending the hematopathology program, which is also very good, but I think it doesn't bring the kind of innovation that we see here at this forum. So thank you very much for your presence, and I think for the next programs, maybe we should try to bring more people into this forum, and because molecular pathology forms kind of the basis for all the pathology now. So thank you, Dr. Mina. Would you like to say something?
I think I second in that. Molecular pathology is a tool that is applied to all the disciplines of pathology, be it microbiology, hematology, histopathology, chemical pathology, and of course molecular biology. So we will encourage not only people from molecular, but from histopathology, hematology, microbiology to join the forum, this Association of Molecular Pathology Pakistan, so that we can do more high cutting-edge technologies, use cutting-edge technologies to do our research as well as diagnostics. So we welcome you all to this forum, and we will have Facebook presence, we will have the registration done, and then we can take more people into it, inshallah. Thank you.
Assalamu alaikum ji. I'm Dr. Shavana Shahid. Today we are going to start this translational research in molecular pathology session, and our first talk would be by Dr. Samina Ijaz, and she would be presenting the unique role of tumor suppressor gene in acute myeloid leukemia. And Dr. Samina Ijaz is Associate Professor, Institute of Biochemistry, Biotechnology and Bioinformatics, the Islamia University of Bhavalpur. And she has been received 2 funded international research project funded by HFC, PSF and NSFC China. And she had a vast experience of research, almost 10 years, in the field of molecular biology of cancer. And she had more than 40 published research articles with the impact factor of 120. So I would like to invite Dr. Samina Ijaz to present her.
Bismillahirrahmanirrahim. I'm here to present the unique role of some tumor suppressor genes in acute myeloid leukemia. And when I planned this research, I asked the question from the understanding of the leukemia and going up to the prevalence. Leukemia is one of the hematological malignancies, and among leukemia, my focus was acute myeloid leukemia, AML. And why leukemia? The answer gives the prevalence of this disease, that 15 to 20% of AML cases have been reported in children and 80% in the adults. And here, leukemia is the 15th most commonly diagnosed cancer, and it is the 11th leading cause of cancer mortality worldwide. And this data is of 2018. And we can study the statistics that in males it is 6 out of 1, and among females it is 4 to 3. And same is the case of mortality, that it is higher in males and it is less as compared to the females. And prevalence in Pakistan, again the same story, same trend that has been observed the worldwide. That in cancer, unfortunately, due to the existence of the cancer registry program, we don't have any authentic, validated, published literature. Although I know that Shaukat Khanam has been working on this cancer registry program, but in the published literature, we do not get enough for the prevalence in Pakistan. And I have mentioned some statistics and figures here regarding the prevalence of this disease in Pakistan. Many of the risk factors and many environmental risk factors, and if you go to the genetic risk factors, that is the particular interest of my lab, here we can observe that story begins with the DNA damage. And the DNA damage response proteins, they have got central role in the management of the, you can say, defense system that prevents the development of the leukemia. And here, if we go to the age, if we consider the age and various epidemiological factors, you can see here that acute myeloid leukemia, this is the early or childhood cancer, and same, and here in case of the CML, that is chronic myeloid leukemia, you can see that it is the disease of the old age. And when we go to the genetic and molecular landscape, we can observe that many mutations in many genes, and these mutations may lead to the altered expression of the genes and genetic basis. Then people have been trying to investigate the genetic basis using the cytogenetic analysis, cytogenetic abnormalities, and cytogenetic abnormalities have got the main focus of the studies because they have been observed in a very high proportion of the patients, that is the 55% of the patients, they are associated with the aberrant proliferation, and you can say that leads to the problem in the differentiation of hematopoietic cells. So cytogenetic profiling and the molecular profiling people are doing. And when we go to the genetic factors and we consider any of the cancer, regardless of the leukemia, we consider the two categories of the genes that are oncogenes, which are if activated, then the problem will be the cancer, and the other category of the genes, that is the tumor suppressor genes, which if inactivated, then problem is there. And my focus or my interest was more for the tumor suppressor genes, that what if inactivates, it leads to the development of the cancer. So it means the tumor suppressor genes, they are working as a protective shield, and that protective shield, if it is lost, then the problem arises. So among the tumor suppressor genes, I started studying few of the tumor suppressor genes, BRCA1, BRCA2, and P53, and then PTEN, and you can see here the type of cancers that all these genes have been associated with different types of cancers, and I found that BRCA1 and BRCA2, they were associated with breast cancer, and same was for P53, and rather the story was more interesting for P53 because it was mentioned that in the literature that 50% of the cancer cases, they have either mutated P53 or defective P53 gene, so that leads to the altered expression and synthesis of the altered protein. So I selected out of these tumor suppressor genes two for my interest because P53 and BRCA1, and both of these genes, it is mentioned their location on the chromosomes, both are located on the chromosome 17, and both these genes, they have got very important role, that they are the DNA damage repair proteins. And so they make it possible that whenever there will be any threat to the DNA and the DNA is damaged, so they will make sure that that damaged DNA is repaired with the full efficiency. So they are working like a defense system. So if defense system is in danger, of course the whole genome is on danger because they are referred as the guardians of the human genome. They make sure that the genome remains in its integrated form, it remains in its original normal form, so that the normal life can proceed. And P53, I have just mentioned here the domain structure, protein structure, so that you can get an idea that both these genes, they work as DNA binding proteins, and there is large part of this protein that is encoded, that is the DNA binding part. So this is the part that these proteins use to bind with the DNA at the specific position and then maintain the transcription of many other genes. So many other genes are dependent upon these two genes. So it means considering these two genes means that we are considering all the downstream genes which are being regulated by these two genes and the proteins. This is the signaling pathway. And here, as I mentioned, story begins with any risk factor that cause damage to the DNA. And we can see here that whenever there will be the damage, so few of the proteins, including ATM and these, they will recognize the damage, and then they invite many other partners. And if we go to the next figure, we will be more able to understand that suppose whenever there is any damage in the DNA, this damage is first responded to this damage is ATM, not the money machine, but a gene that encodes a very valuable phosphatase ATM. And ATM recognizes and then it invites it interacts with the other proteins, and finally the message is received by the P53, and this P53 immediately it will halt the cell cycle so that it will ensure that the defective DNA is not transferred to the daughter cells. And then meanwhile it will communicate with the DNA repair machinery including BRCA1, BRCA2 and many other proteins where the DNA damage is, you can say the complex, this is mega complex, many proteins are involved and BRCA1 is one significant member of that complex. So the DNA damage repair machinery tries to do the job. But if the damage is irreparable, then P53 has to sense that situation and it has to invite the apoptotic machinery that this damaged DNA damaged cell with the damaged DNA, it must be cleared off from the body. And then if the damage is not, if P53 is defective, it cannot halt the cell cycle. It cannot communicate the DNA repair machinery. It cannot judge the situation that this damaged DNA is going to transfer to the daughter cells, and it can appear it can lead to the leftover mutations in the daughter cells, and daughter cell can be converted into the problematic cell, and that problematic cell can be the cancer. And here, just to give you an idea of that all this that we have seen in the previous figure, and then you can see that how many genes are dependent upon this P53 because it is acting as the transcription factor, and these are the target genes. And here I have made the different categories. So each category you can see apoptosis defective in cancer, growth arrest, if it is defective, not at the particular, it leads to the cancer. So these are some figures that I have added just to give you an idea that story is not simple. Story is not the matter of only the two genes and two proteins. This is basically too many mega protein complexes are involved, but I'm just focusing on today. My research lab has been investigating the role of from ATM and many other partners which are supporting the P53 and BRCA1. But I am today here to present only BRCA1 and P53. So BRCA1 and P53 associated mutations, I went to the literature, and this was 2017 when I was planning my research, and I observed that many mutations were available for BRCA1, 2 and P53, but for breast cancer and other cancers, but not the leukemia. Nature of these mutations was very diverse, including missense, frame shift, and the supply side mutations. So this was very exciting moment for me that when I planned this study. But if this will be the dishonesty if I'm not giving the first report that was published in 2020, you can see we planned this research earlier to that because at that time there was no report for the P53 involvement in the leukemia worldwide as well as in Pakistan. But this study reported few of the mutations of the P53, and they got advantage of that. Our paper was in the process, and so but they first published, and so you can say that P53 role was in worldwide population, but not in the Pakistani population. And all the mutations, you can see here that majority of the mutations, they are in the DNA binding domain. because this is the domain that is central to its role. If it is defective, this protein is not translocated to the nucleus, it cannot bind with the DNA, and it cannot repair the damaged DNA. So, and majority of the mutations, they were affecting the DNA binding domain of this protein. And same was true for the BRCA1. And here the exon 11 was the hotspot. And that was, most of the mutations were reported. So at that time, I identified this gap as to a question to go for, because there was no information regarding the role of these genes in leukemia and particularly in Pakistani. But because I'm claiming today, so I can say in Pakistani population. But when I planned this study, it was not even a single report worldwide regarding these genes for the leukemia. And why I thought? Because I have mentioned their role, and I thought that it would be more appropriate if I can go for these genes, because they are the most suitable targets. They can be used as biomarkers of leukemia. Why? Because they have role in growth, differentiation, and apoptosis regulation. And we hypothesize that we can, due to population-specific genetic differences, we can find some unique mutations and expression profile. We can identify some specific isoforms in Pakistani leukemia patients. And these were the aims and objectives. We started studying. We first optimized a protocol that can be used for the extraction of DNA from very low volume of the blood sample. And this was just like, you can say, 250 microliter of the DNA, and using the manual method, not the kit method. And then we planned primers, and we went for the PCR amplification, then sequencing, and then elucidation of the functional impacts. Here I have mentioned the research methodology. We planned the dry lab experiments, where bioinformatic analysis and statistical analysis, wet lab experiments, collection, mutational analysis, expression analysis, and then bioinformatic analysis was divided in two parts: pre-wet lab phase and the post-wet lab phase. Pre-wet lab phase, where I first searched the available databases, that what is known there regarding these genes, their isoforms, their mutational profiles, and this information I used for designing of the gene-specific primers for PCR amplification. In the post-wet lab phase, when I received the data of the DNA mutations, then I analyzed to do the comparative analysis of these mutations, and then the theoretical predictions regarding the functional impact of these mutations. And then mutational impact, and here at the translational level, at the transcriptional level, and then for the transcriptional level, we went for the PCR amplification. For the protein part, that we extracted tissue proteins and analyzed them. And these are some of the techniques which were used for the to document the aberrant expression of these proteins on the translational level. PCR program I have mentioned. If anybody will be interested, I can share this information. And then it is important that we design the primers here for the mutational hotspots. And then we went for the PCR amplification. And surprisingly, I would like to mention here that we were able to find no result in few cases, and where the meaning was that either the target region is absent, or there is problem with the target region, so that the PCR amplification cannot take place. So this is the gel of the BRCA1, and then positivity ratio was calculated. Expression of the mutational extension of the mutational analysis was done again for the post-bioinformatic phase, post-wet lab bioinformatic phase, and I am here to present 5 cases. This is the case 1, where I am mentioning the mutation, and you can see here that mutation, location of the mutation, and interesting phase is that when you go for the functional impact, here the DNA binding domain is truncated. So protein is truncated and it is losing its nuclear localization signals. And when it loses its nuclear localization signal, this protein will not be translocated in the nucleus where it has to play the role. So same is the case here, and you can see wild type normal protein and the mutant protein here. The case number 2, and you can see here again many mutations in the same patient, but all in the DNA binding domain. And what is the defect? Defect is that these target protein, what is DNA DNA binding domain is there, but it has lost those essential amino acids which are essential to interact with the DNA. So, and then I determined what is the impact on the 3D structure, and you can clearly see that localization is cytoplasmic in case of the mutant protein, so it is not translocated to the nucleus. And here in case of the, these are few other mutations, you can see here that complete DNA binding domain is lost. And then same is case of the localization defect, that is it remains in the cytoplasm, and this is some unique mutations which we only observe in the Pakistani population, and internationally they have not been reported even till date. And these are some of the mutations in the BRCA1, and same is the story. And you can see here that they can bind with the DNA wild type proteins profile and the other protein profiles they are given. So you can see here that there are many defects in the structure, there are many defects in the structure localization, but in case of P53 they are more significant. In case of the BRCA1, although its role is being affected, but its location is not being affected. And here this is the conclusion that justifies that normal P53 normal process, but if mutated, no BRCA1, no P53, and increased risk of leukemia will be there. And there is need to confirm these functional impact of these mutations experimentally, and my lab is working on this, and we have collected the data, and we will be publishing, and it is under process in scientific reports. And this research has made the impact in quantifiable terms. I can claim that we have provided an update on the nature and type of mutation.
Sorry, we are running out of time. If you could just conclude.
And take home message that we need to reconsider the role of both of these proteins and genes in AML. Thank you very much. These are some of the references, and I would like to acknowledge the patients who participated in this study, my students, without which this work cannot be completed, the lab staff, and two of my mentors who are present here, Dr. Ramina Kazi and Dr. Suhail. Thank you very much, ma'am and sir, for being here and for teaching me all this that I have been able to present here today. Thank you very much.
Thank you so much. This was an excellent talk and very important one. I think everybody has a lot of questions, and I also have a lot of questions, but maybe I will utilize the opportunity of the lunch break to ask those questions. So maybe we move on to the next presentation.
Thank you, Dr. Samina, for such a good insight with P53 and sharing the mutations in Pakistani population. Well, that's wonderful work. From here, we will move forward to our next speaker. Our next speaker is Dr. Amreen Fatma, and she would be presenting genome editing tools, insights into translational breakthroughs and future challenges. Dr. Fatma is assistant professor at Khan University, and her research interest is to understand the genetic aspects of rare disorders, all the way from the identification of causative genes to pathophysiological mechanisms with perspective of therapeutic interventions. Her expertise are in genomics and disease modeling using 2D and 3D iPSC technologies, CRISPR-Cas9 genome editing, RNA sequencing, and high throughput data analysis. So I welcome to the podium Dr. Amreen Fatma.
Assalamu alaikum everyone, and thank you for the nice introduction, and thanks to Dr. Natasha and all the organizers for inviting me. Okay, so I have nothing to disclose. So we'll start with the human genome. So I call it Book of Life. It have all the instruction, everything, how we perform, how even we behave, and everything is in this book. So during last 2 decades, now we are able to read this, and we are able to understand this book And of course, there are many things in this book that leads to disorders, and sometimes just one single change in this book, in these genes, or like any of these genes can lead to very lethal diseases. I put some examples here. For example, this C2, if the C is replaced with the T, yes, so it can lead to this early aging. Similarly, if there are many mutations in beta thalassemia, but there are point mutation, thus one replacement of G into C can cause beta thalassemia, and then similarly, mutation like single nucleotide variation called phenylketonuria, and sickle cell disease, that just one mutation, just one amino acid change can lead to this disease. So there are more than 10,000 such disorders that we have been we were able to understand during the past 2 decades. So if you would have asked, or we would have been asked like 10 years ago, that can we revert these back into normal, that probably was a science fiction, and we would think like, no, that's not possible. But now it is, and during next 20 minutes, I'm going to tell you something about these genome editing tools, work that people are doing, and some that we are doing at Aga Khan. So I'm sure you have heard about the CRISPR. So CRISPR is not something that just we invented. It was pretty much in the nature. So it was when in 1987, when the bacterial genome was sequenced, so scientists have found that there are some regions, those are not from the bacterial genome, so they are just sequences, and later it was identified that these were the viral sequences that become the part of bacteria. So basically, CRISPR is an adaptive immune system that bacteria have adapted to kill the viruses. So just briefly, because we don't have much time to explain all of these things, but there are many wonderful videos and things on YouTube if you want to understand CRISPR. They probably will explain better than me. So when the virus attacks, so bacterial protein, Cas1, 2, they actually take some part of the viral genome and integrate into the bacterial. So when again there is attack by the virus, so of course these proteins are transcribed into form of RNA. Sorry, these regions are transcribed into form of RNA, and then the Cas9 bind and make a complex, and that's actually that cut the viral genome. So scientists actually started thinking if we can use this system and to edit something in the human genome, and then they actually use this. So there is this protein, Cas9, but there are many versions of Cas protein available. So there are certain domains of this protein, and then this is the system that we call guide RNA, and it have two things. So one is that this part, the green part that binds with the Cas9 or the Cas protein, so this is constant. So this red part is something that we design, and it's just making like similar to making a primer. So it's just you have your region, and then you find and then you make this sequence according to your region. For example, the whatever gene that you want to edit. So when there is a complex, so basically then it's a complex, and it bind and make the bind to the DNA and make double strand break. So there are many domains in this one, and now scientists have started playing with these domains and making it to different version of Cas9. For instance, there is a version of Cas protein available that we call dead Cas9. It still binds to this one, but it is unable to make the double strand break, and we will discuss in later slide what is the use of such protein. Okay, now when there is a binding and double strand break, it's kind of an emergency situation for the cell, and the cell machinery will try to repair it, right? There are two mechanisms. So one is this non-homologous end-joining. They will just repair it, and this is an error-prone method, and that leads to knocking out genes. So in this case, this is like a more efficient method, and we can delete the gene by using this method. On the other hand, if we are interested in making a specific change, if we are reverting those single nucleotide mutations back into their original form, so then we can also provide, beside these RNA and protein, we can actually provide another piece of instruction that we call repair template, and that instruction will guide the cell machinery to repair that system according to our instruction. So these two ladies got Nobel Prize in 2020, and this was one of the quickest Nobel Prize in the field. I would I put this guy's picture here because his work is like amazing. And after the CRISPR discovery, David Liu from Broad Institute, so he actually played with the guide, those Cas protein, and made into dead Cas9. And then now there is another method of editing, those a lot of people are using, and this is called base and prime editing. So base editing is that it will bind the similar way, but it will not cut. Instead, it will modify the base, that correct the base. So just to make the simple comparison of these three techniques is that CRISPR is like scissors. It will go and cut. Base editor is like eraser and pencil. So it will erase, and then of course it will write the right base there. And then this is a bit more complicated, yet it have like huge potential, and now everyone is using it because this is something that we kind of program, and we can correct all kind of mutation, whether it's inversion, translocations, single nucleotide variation, whatever is possible with the prime edit. Okay, so when it comes to application, probably it is not possible for anyone to write all the application these techniques have. So I mentioned like now we are using it for every disease, almost every disease, whether it's when we want to know the function of these genes, like for example, if we are interested that what this gene is doing in brain or in any other organ, so then we can actually make similar kind of mutation that we find in our patient and look at the function of those genes. But I will focus mostly on the other part, that is how we can treat the disorders. So to look at the function of genes, this is like how we do it. I put one of my paper here, and the reference is there, and then if you are interested, you can look into that. So again, because of the time, I'm just kind of running like fast. So this is a gene, and this is like we targeted these two exons, and we made this two deletions. And one was 16 base pair deletion that was again leading to stop codon, and this was a larger deletion. And here is a Western blot that you can see the wild type have a protein, but both of our knockouts are not expressing the protein. So now we were interested to know that what this absence of this gene is causing, because our patient have epilepsy and intellectual disability. Similarly, we knock it out in the stem cell, made it into neuron, differentiated into cortical interneurons, and look at the function of those. So then again, not going into these complicated graphs and things. If you are interested, please go and look at this paper. Okay, now we are going to talk about some fancy stuff in terms of disease treatment. So this paper was published in New England Journal of Medicine in 2019, and this was one of the first paper that came as a treatment option of CRISPR for rare disorders, and this was for beta thalassemia and sickle cell anemia. So the strategy that they have used was very intelligent. So we know that there is a fetal hemoglobin, and then there is adult hemoglobin. And when the baby is born, then there are certain genes. For instance, this BCL11A is the gene that actually tell fetal hemoglobin that now you are not needed anymore, so stop it. And that gene is BCL11A. But because in the thalassemia and sickle cell anemia, the patient adult hemoglobin or beta hemoglobin is not there, then what they did was they actually removed this gene. So this gene is suppressor, right? So if this gene is not present, it means the fetal will be available, and then that will kind of make the patient hemoglobin available and make them transfusion independent. So that time they started trial using this technology, and they have developed. This is quite common. You design something in lab, and then you develop a company, and then of course you go further on that. So they developed these two companies, CRISPR Therapeutics, another one. So they started clinical trial now, I think. So they have data of 31 patient and a follow-up data of at least more than a year. And then there is another company that is Beam Therapeutics using base editors, and that is from David Liu's group. There is another company, and that what they are doing is they are not making mutations, correcting every mutation. Instead of that, we know that beta globin gene is not a huge gene. So what they are doing is they are actually replacing it with the correct copy. So it will be mutation independent. Whether the patient have IVS15, frameshift 89 or any other, that strategy will work for that. Just day before yesterday, the UK have already approved the world's first CRISPR-based medicine for the for the sickle cell anemia. So this was also submitted in first week of November to FDA, and hopefully we will get the FDA approval. This is like a huge breakthrough in the scientific for the scientific community. Okay, the base editor. This is the progeria that is the early aging. I think my video will not work. Oh, it is. So this is the mouse data they have shown. So basically, this is a mouse showing early aging because they made it a knockout mice showing the disease. And this was not moving. It was showing all sign of progeria. But when they treated it just with the single injection, you can see now it's pretty much working and functioning. So, and they actually took the follow-up data for maybe 500 days for this mouse, and it was just for the one injection, it was working like a magic. So again, they have started clinical trial for this one, and this is also from Broad Institute, David Flew's group. Okay, so a lot of people work on leukemia and cancer, and you know that how big, and especially the drug, the resistant kind of leukemia. Again, I'm not coming from a cancer background. Maybe I say something wrong, but okay. This was another wonderful trial that was used first time to use CRISPR-based, base edited CAR T cells to treat leukemia patient in UK. And this was done in Gauche and UCL, and this was done by Professor Wasim Qasim. And if you are interested, the paper is again published in New England Journal of Medicine this year. Wonderful, amazing work. CAR T cell again have a huge potential to treat a lot of cancers. But if we couple it with CRISPR or genome editing tool, make some modification in those cells to fight against those cancers. So this is again a wonderful thing. There are 3 papers published just this year in American Journal, HSG Advances in Nature Communication. By the same group, they actually had wonderful model of using prime editing to treat phenylketonuria, and they have got a huge funding now to go into clinical trials. So again, these papers are there. So this is something that I'm just telling you that a lot of advancement is done in the field and people are working. I think so like 2-3 days ago, this was again something that is not only for rare disorder, but there is a lot of hope for common disorder or cardiovascular disorder. The first clinical trial started for PSK9 gene. So if we delete this gene in liver, so we can protect from the cardiovascular disorder. So this is again something huge, and we know that these cardiovascular disorders have one of the largest cause of the death in worldwide. So this is something amazing. Okay, now I'm switching towards what we are doing at AKU. So we have received this Grand Challenge Fund from Higher Education Commission, in which Dr. Afsar is PI and I'm co-PI, and we have a couple of other people working on that. So we started with the BCL11A. But instead of using the region that they have used in the CRISPR therapeutics, we are targeting different regions. So now we know that these computational tools, how important they are, and to understand the genome better. So we have looked in the different kind of domains and DNA binding things, and we have found that, okay, they have targeted the exon 2, and if we target the exon 4, they have the better binding and of course the better efficiency. So we might can induce fetal hemoglobin more efficiently. So to work or to look at the function of this one, so this is some of the data that I'm showing. So we have done this in case 562 cell line just to validate that our system is working. But then we also did in CD34 hematopoietic stem cell that we derived from the patient. And then we deleted these BCL11A from the exon 4. And this is our knockout band. And this is like, this might explain it better. So here we have done both ways. So we have done what previously was done by targeting the exon 2 zinc finger domain and by targeting the 4. And as we expected computationally, we can see that our fetal induction in in the region that we are targeting was much better than that have been previously done. So this might be an efficient way. So we also show that on Western blot as well. So, and now we are working on going towards in vivo experiment. So this is again a confirmation we did on HPLC. So here is the control for both. And here you can see that this is the fetal hemoglobin induction by the exon 2 deletion, and this is the fetal hemoglobin induction by exon 4 deletion. And you can see that this is much more efficient than what have been done in exon 2. Okay, so now we are doing some in vitro model further to go into animal and to the sickle cell anemia and beta thalassemia mouse model. Recently, we received another grant from Welcome League, and I just put these pictures here, not just to show you that how cool we look in this picture, but okay, Dr. Afsan have expertise in leukemia and drug discovery. Dr. Salman Kirmani is a clinical geneticist, board certified clinical geneticist. I work with genomics, gene editing, and stem cell. Dr. Irfan have a wonderful expertise in computational as well as genome editing, and Dr. Fawad have expertise in delivering these methods by using nanoparticles and exosomes. So now this group have different expertise. So it's not that the job of one person can do all these things. So I just put it here because, and of course we have a lot of postdocs and PhD students and master's students that all are helping, but everyone have different expertise, and now our aim is to develop the druggable gene editing tools for beta thalassemia and sickle cell anemia. Okay, so this is our approach. So I have just briefly put like the whole thing here. So we are using both the patient-derived hematopoietic stem cell, that is CD34 positive cells that we isolate by using the magnetic sorting. Another approach that we are using is we are taking patient skin cell, or we are driving fibroblasts from the patient skin, and then we are reverting back those into induced pluripotent stem cell by using 4 Yamanaka factors. And now both of our models have been done. We have fully characterized these. I haven't put like all those graphs and things here. Okay, next thing is that we are optimizing now prime editing. So as I mentioned that prime editing have left or off target. Another novel thing that we are trying to do in this one is that we are immortalizing it and making it kind of, because one of the problem with the CD34 positive cells is, of course, they have a half-life and then they will finish after some time. So now what we are doing is, instead of adding an oncogene, we are making it a transient immortalization, and we are optimizing those. And once those will be done, then we'll go to in vivo model. And after in vivo model, ultimately our aim is that we make this in a system that is we can inject into the patient and it directly go for the genome editing. So as I said that we have a person who is well equipped with all these delivery methods. So we are approaching different type of delivery method, and then we'll see like which one works the best. So for instance, we have designed a peptide, a small kind of small peptide, and we are that target only the hematopoietic stem cell. Another thing we are doing is we are using CD117 antibody, and that will also directly target the hematopoietic stem cell. We are exploring these other methods because we want to have more efficient method that go directly to your target. Why we need this? Because I will actually explain in a little bit. Maybe I can go there directly. This is my last slide. Don't worry. Okay. So why I said the delivery methods are important? As we have seen in this last presentation by Dr. Samina, that there are, genes that doesn't work in silos, right? So gene have interaction with other, and then of course if we are removing something from the body, and it might have another effect, right? For example, this BCL11A is also have some effect on the brain function. But now the target is that we remove or edit these genes in a specific tissue. That is, for example, we are doing in hematopoietic stem cell, not in the brain or other organ. So these delivery methods are needed to deliver our target to directly the cell population that we are interested, whether it's liver, whether it's kidneys or bone marrow or whatever the disease, depending on the disease of interest. So another thing is that we need to have better gene tools that have better gene editing efficiency, that they can actually correct the maximum population into the normal. Delivery method that I already have covered, reduce off-target effect. For those who doesn't know what are off-targets, it's basically beside where we need, these system can go and do editing in other places, and we do not know, we do not want that. And that's why we are focusing on better the one that does not have off target, for example, the prime editing and base editing. I think the ethical issues are one of the most important thing in this. These are fascinating discoveries. We all want to treat these disorders. We all want to treat these patients. But now there are many things. For example, we know that, okay, we can edit the eyes color, we can edit all those things, but do they are needed? Probably not, right? So it's very important to look and have the regulation that what is needed, what is done. And if you remember, like in 2019, there was this one kind of controversy that came from China, that this guy have actually made the first human genome edited babies, genetically modified babies, and it was just deleting the CCR5 gene from the embryo, just to and of course he went to jail. And but it's kind of important to regulate these discoveries. And again, this is kind of a scissors, but it doesn't mean that we can cut wherever we want. We need to know the function of these genes first, the tissue, the right delivery method, and all those things. And of course the cost. And cost is huge, right? These are like $1 million treatment, and we probably, our patient cannot afford it. Though we have the highest burden of genetic disorder in our country. For example, if we even talk about beta thalassemia, it's globally highest in our region, but we cannot treat that. But then, of course, how we can overcome this inequity, and how we can bring these solutions, or we can bring these technologies into our country, and to make collaboration between clinician, researcher, and of course pharmaceutical industry. So that's one needed, and of course the regulatory challenges that we have to look into that. That's it. Thank you very much, and I would be happy to answer your questions.
I think maybe that was again an excellent presentation of some innovative work that's being done in Pakistan, and also very relevant. So I think maybe we can again utilize the opportunity of the lunch break to ask questions. And thank you so much.